So, good morning, students. I welcome you in this new session of second semester, and you have already studied the chemical dynamics in M.Sc first semester, and in second semester, course number 9, there is a paper. You can call it. It is an extension of chemical dynamics, part 2. And in this chemical dynamics, basically we will study two important aspects of chemical kinetics. One is fast reactions. I'll explain what is fast reaction and what is the difference between the conventional reactions and fast reactions. And then, of course, I'll explain the different methods available for the study of the fast reactions. It is a very big chapter. So first I will explain today, I will explain some of the fundamental aspects of the fast reactions. Another important aspects of this course CH9, that is called unimolecular reactions. There are many theories available in the unimolecular reactions. And because we will study the fast reactions, and under this fast reaction, we will study the molecular reaction dynamics and how to probe the transition state. Okay, so today I will concentrate only on the fundamental of the fast reactions, and then of course some derivations and other things. As you know that whenever I teach any subject, I must tell the student what are the books which are available in our library, and you should refer some of the books for detailed information of the fast reactions. As you know, during your first semester, I do believe that you have already referred the most important book of chemical kinetics, that is K.J. Ledler. And in second semester also, you will find many topics will be covered by this book K.J. Ledler. And I have also referred some of the derivations from the K.J. Ledler. Now suppose you want to learn details information of the fast reactions. So I must tell my student that you must refer only Indian books available for the fast reaction. Only Indian book available for the fast reaction. I think there are a large number of copies are available in our Pandit Ravi Shankar Shukla University, Raipur. That is Chemical Kinetic Methods by Professor Chetty Kalidas. He was a professor of IIT Madras. And fortunately, he visited this department long back and delivered a series of lectures on this fast reactions. So this book is very good. Now in addition to that, there are some monographs are also available in our library. If you want to go into details of the fast reaction, you can refer all such type of books. And as you know that, because now if you see the modern new education policy, there is a term that is called the research oriented teaching. And I have many times I told you that there are many good American Chemical Society journals, Royal Society of Chemistry journals are available for the student. So if time permits, you must read some of the research papers so that you can understand. The language is also very simple, because whenever I am talking about the fast reaction, fast, so you must know, sir, why it is called the fast reactions. So if you read through this, if you go through this very simple article written by a great scientist, Kruger, in Chemical Society Reviews, long back, I think it is a very old paper, and you will find the actual difference between the slow reaction and the fast reactions. Anyway, so let me start with the kinetics of the fast reactions. Now, the first very obvious question. Obvious question means that because you have already performed, I think 2 or 3 chemical kinetics experiment during your M.Sc first semester, and you have measured the rate constant as well as the half-life period, and I think that you got like 1 into 10 to the power minus 3 second inverse 1, like that, or 1 hour half-life, like that. But I think you have not aware of, just you imagine, you just compare with your conventional reactions as well as some abnormal type of reactions, where you will get the rate constant or half-life 10 to the minus 10, just beyond your imagination, 10 to the power minus 15, 10 to the power minus 9, like that. So basically I can divide this kinetic time range in less than 1 second, or 10 to the power minus 10 or 10 to the power minus 15 second. That is, sometimes it is called the femtosecond, picosecond, nanosecond, like that. So let us give one simple example. You can see it there. Huh? So why I am giving this example? Because you can understand the things. Hydrolysis of methyl acetate. You just first you control and then you can see it. Most of the are reactions. So it is a very common reactions. During your B.Sc or M.Sc you have performed this experiment. It is because we need simple burette and pipette, and without any problem you can determine the rate constant. So it is an approximate value. At room temperature or at any temperature, we are getting 1 into 10 to the power minus 3 or like 2 into 10 to the power minus 5, 4, like that. It depends upon the temperature also. So this is called slow reaction, because the half-life period is around 700 seconds, like that, 10 minutes or like that. So that is not at all fast. So by simple titrometric method you can determine this reaction, or even if you are doing the saponification of ethyl acetate using the conductometric method, that means you are measuring the conductance of that solution. So generally what we call slow reactions which can be measured with the help of simple conventional technique, and half-life is more than 1 second. 20 seconds can be also, because with the help of simple watch, or you can monitor this thing. Just now I am giving you the second example. You take the ferric chloride and HCN solution, and immediately fraction of second there is a red color. So my question is that, will it be possible for you to study this reaction with the help of burette and pipette? No. And if I ask you, can you tell me most common first reaction in whole chemistry? You will be surprised to know, and there are, in fast reaction technique, there are 3 or 4 Nobel prizes in these things. And you always remember this reaction. That is, the formation of water is one of the great example of fast reaction. And a great German scientist, Manfred Eigen, awarded Nobel Prize in 1967 for this water formation reaction. Now just to compare the hydrolysis of methyl acetate, rate constant is 1 into 10 to the minus 3 approximately, maybe minus 5, maybe minutes, maybe second, and at room temperature the formation of this K1 forward reaction. Of course, it is a second order reactions, and you just see, imagine the rate constant is 1.4 into 10 to the power 11. You just can you imagine 10 to the power 11 liter mole per second. It is a second order reaction, and of course the dissociation of water is the slow reactions. It is around approximately 2.5 into 10 to the power minus 5 second plus 1. Now if I calculate that, just like your half-life period, if it is first order, you know that 0.693 divided by K, like that. Like we can calculate, sorry, I think K divided by 0.693, like that. Or if it is 1 divided by K and A, or like that, initial concentration. So if I calculate these second order reactions, and it is calculation is 0.1 like that, or whatever it is 1, so it will just 10 to the power 11. So they are approximately 10 to the power 11. So you just compare 10 minutes, 10 seconds, and 10 to the power minus 11. So what is the definition of the fast reaction? There is no proper definition of the fast reactions. Reactions cannot be determined, studied, or rate constant cannot be determined by simple conventional method. Because if you just want to mix, say for example, you take one conical flask with this methyl acetate, another conical flask with some acid or like that, isn't it? Then you mix it, and then you mix it, and with half discharge you just take some aliquot and monitor with the help of titration method you determine. So mixing time is not very difficult. But if such a reaction, I am not talking about the formation of water, there are thousands of reactions. But if the mixing time, that means the time required to just mix, reaction is finished. So how do you study such type of reaction? Such type of reaction is called the fast reactions. So therefore, we have to study some special method for studying such type of fast reaction. And it is nothing, because it is a mechanical device. Just you think that now you are doing manually, but suppose you have some very sophisticated equipment, and automatically you are forced to mix this solution in a fraction of a second and monitor their properties. That is the technique. That is called the techniques of the fast reactions. Now, as I told you that normally milli nano chemistry, millichemistry, micro. I'm talking about this range. Half-life period is like suppose 10 to the power second. Actually second inverse. I'm just example. So fast reaction actually less than 1 second, 10 seconds or like that. 10 seconds. So now it is also possible because there are two important Nobel laureates I will discuss. I think Nobel 1999, that is called the femtosecond spectroscopy. When I will teach the molecular reaction dynamics, I will discuss some of these things, but fortunately we have not included ultra fast technique in your course. So don't worry about this femtosecond spectroscopy, transition state spectroscopy. And Professor Ahmed's well awarded Nobel Prize, these are all ultra fast technique. And you will be happy to know that up to 10 to the power 15, no problem. But now, actually the movement of the electron and the velocity of the electron can also be studied, that is called the attosecond, 10 to the power minus 18. And in 2023, Nobel Prize awarded for this attosecond spectroscopy, of course not in chemistry, but in physics. Then picosecond, femtosecond, nanosecond, like that. Yeah, I am just giving this spectroscopy. That means these all are called actually ultra fast technique, and this is the fast reaction. So don't worry. Now with the help of very sophisticated reactions, sophisticated technique, fast reaction can be studied. Now the second question. Suppose all the methods are based on the types of the reactions. Suppose you want to study a particular reactions whose half-life is suppose 10 to the power minus 1. Okay. Another reaction is half-life is 10 to the power minus 9. So what you will do? For 10 to the power minus 1, there are certain technique. For 10 to the power minus 9, there are certain technique. 10 to the power minus 12, there are certain technique. So depending upon the half-life period or nature of the reactions, the techniques have been divided. Depending upon the half-life period, the techniques have been divided. So because you are student, so I will start from very simple technique, then gradually I will go to the NMR technique and last. So this is in your course. Okay, is it clear? What is fast reaction? How fast reaction is divided? Very simple. It is not at all difficult. Now sometimes in examination, as I told you that it is up to you. My basic intention is that I teach all the fundamental things. Maybe important for the university question, like that this year you have not seen a single question on the rate laws. Okay, but it's very simple question. Chain reactions, inactivated complex theory, primary salt effect. The question is very simple, but sometimes maybe. So don't worry about these things. Sometimes a short question can be asked that give the 3 or 4 example of fast reaction. So I have taken from the literature some of the most important example of fast reactions with their rate constant. These are all approximately. I have taken from some books, some research papers. So basically, number one is always formation of the water reaction. Actually, we should give the formation of the water, formation of the ammonium ion, like that. And you see that the protonation of a very weak base, that is also very fast reaction. And most of the biological reactions, like enzyme catalyzed reactions, photochemistry of the vision, you will learn in the 3rd semester in photochemistry. Photochemistry of vision is also one of the best example of the femtosecond spectroscopy. Conformational analysis from one beta-rhodopsin or meta-rhodopsin, there are a large number of changes, is also a fast reaction. simply some oxygen hemoglobin reaction, most important reaction, you know. Without oxygen hemoglobin reaction is also an example of, and even they are able to calculate the rate constant. You see, like that. So, say 10 to the power 10, 10, 10, 11, like that. So, these are some example of fast reactions. So, photochemistry of visions, photosynthesis reaction, enzyme catalysis, the protein synthesis, there are large number of reactions are very fast, and biological process, you know, there are a large number of processes, but we will not discuss, because we are here, we are studying the types of the fast reaction, the technique of the fast reactions. We will not discuss the biological application of the fast reaction, but you must know that most of the biological reactions are fast reactions. So therefore, large number of scientists are working on this biological reaction with the help of fast reaction techniques. Second, most of the environmental reactions are also very fast. So therefore, you need some very good techniques. Fortunately, in my next class, on Thursday, I will show you the stop flow technique. When I got a DST research project for studying the fast reaction, protonation constant, so we purchased this stop flow technique. In next class, I will show you. This machine is very simple, so you can understand the concept is very simple. Now, before I start it is again my style of teaching is that I always give the history. Although nobody will ask you in the examination that write the history of that fast reaction, no. But you must know that the importance of the fast reaction. There is a large number of Nobel Prize on these things. I will show their photographs, so you can understand the importance of the subject. Not for the examination, because always I give this example, the water reactions is one of the most important, and I will solve some numerical problems in your second class or the third class. And similarly, the formation of the water and formation of the heavy water. If you go to my lab, you will see that heavy water. It is just like water, and it is produced. I cannot purchase the heavy water from anywhere because it is from the Bhabha Atomic Research Center. So we do purchase long back. We do lot of work on the chemical kinetics. I purchase heavy water, deuterated hydrochloric acid from the Bhabha Atomic Research Center. So even D and OD, formation of D2O is also a fast reaction. Of course, because is 1.4 into 10 to the 11, it is 8.4 into 10 to the 11. And this is the great scientist, Manfred Eigen, who got Nobel Prize for these reactions. And if you, very nice article, unestimable fast reactions, because every Nobel laureate has to deliver a Nobel lecture, and this lecture is available in the website, so you can go through these things. And along with this Eigen, two other scientists, Norris and Porter, you know, regarding the photochemical reaction. That is also a very fast reaction. I will explain. So they also got so three persons got the Nobel Prize. But these fast reactions cannot be followed. I have already told you that even kinetically by conventional method. And it is quite obvious that if you calculate the activation parameters of hydrolysis of ethyl acetate, it is like, if I am not wrong, 12.8 kilocalorie approximately energy of activations. But most of the fast reactions, activation is negative, even very low, very or less chemical reactions. Negative, very, very low, because of quite natural. So fast, it is very difficult to determine their energy of activation. So that is called the barrierless chemical reactions. I'll teach after 5 or 6 period, because that is the last part of this chemical kinetics. Barrierless chemical reactions is very important. Barrierless chemical reactions. But now I am coming to your course, because as I told you that there are large number of methods are available. Large number of methods are available. But you are also concerned about your examination. You are also concerned about your course content. So according to your course number 9, there are only 4 methods. You have to learn only 4 methods. Not ultra fast. We have eliminated ultra fast. We have eliminated that pulse laser technique. We have eliminated the femtosecond spectroscopy, because that is little bit difficult and that is research oriented technique. Okay. So first method is flow method. Today I will discuss only the flow method. Second method is very important. Again, it is Nobel Prize method, Manfred Eisen's method. Flash photolysis is again important. Again, it is a Nobel Prize method. And you know that NMR, there are many Nobel Prizes in NMR. Unfortunately, we have studied the fundamentals of NMR in your second semester, maybe in first semester. I do not know much about this thing. But here you have to study. Unfortunately, we have a NMR machine in our university. So you can understand that what is the importance of the NMR. Again, flow method can be divided into 2 parts. One is called continuous flow. Name itself is a definition. Continuous flow. Continuous flow. Second is the spelling is mistake, STPPOD, and some slight spelling mistake maybe. Correct it. Stopped flow method. So, and in relaxation also 2 methods. One is called the pressure jump method. Another is the temperature jump method. So there are 1, 2, 3, 4, 5, 6 methods. Now, one fundamental point you should always remember, whether it is a NMR method or relaxation method or flow method, everywhere there must be some vessels, reaction vessels. What is the fundamental theory? That we have to minimize the mixing time. So that means there must be some device. Again I am just saying, again you just 2 reaction, suppose this is 2 reaction vessel. Whether you are performing the methyl acetate or hydrochloric acid reactions, or oxygen and hemoglobin, or ferric chloride and thiocyanide ion, whatever it is, or A plus B plus C. So there must be some reaction vessel A and B. Because it is ultra fast technique, so no question of manual, burette, nothing. So there must be some system, so that, and there must be some mixing chamber, like, so if you understand the fundamental, you can derive, you can write anything. Again I am just repeating these things. Concept is same, there must be some 2 reaction vessels. Only thing is that in place of manual system, you need some electronics, you need some very automated system. It may be a NMR, you know, there is a NMR tube. So there must be some system that both the reactants are forced to flow with that a fraction of second. That is the system. And then automatically mix, and like in your hydrolysis of methyl acetate, or you measure the conductance, you measure the absorbance, you measure the fluorescence, you measure the chemical shift, that's all, with respect to time. So first we will discuss the flow method, but before that I will show you a table so that you can understand the different methods and their half-life. Again, these are the same thing in terms of, it is a tabular form. My students have prepared this table, and as per the course, and this is also same thing, flow method, relaxation set, it is not in your course, photophotolysis is in your course, NMR method is in your course, ultra technique is not in your course, pulse laser is not in your course. So now I just go through the history of the fast reactions, because flow method is the most simplest method, and long back, you just see 1897, E. Rutherford discovered this flow method, and I think he measured a fast reaction, his half-life is 0.1 second. But there are certain modifications. Hartridge and Rafton, you know, is a great scientist. They also studied the flow method, and there are many types of flow method. Flow means, you know, it is just like a simple system, the flow of reactions, flow of the reactions. Sometimes it is called, it is related to some physics and other things. Flow reactor process. No automation is required for that. But because you are students, so you must know the first method. You must know the first method. And then, of course, gradually we will process, and Millikan also discovered. Then, this is the most important, very important, most important. Today I will not discuss this stop flow method. Today I will not discuss this stop flow method. In my next class I will discuss the stop flow method. So it is actually discussed, derived by B. Chance in 1940. Because there are some problems in the flow methods. There are some problems in the flow methods. A very large amount of chemicals are required. But in stop flow method, very small amount of chemicals are required. A very small syringe like that. And you will get the accurate rate constant. Fortunately, we have this stop flow technique in our system, in our department. So I will show you. This is a very small machine. That time it was cost is 4 to 5 lakhs. Long back I purchased from a, I think, DRD or DST project, like the, for studying the protonation constant. So this is the first history of the flow methods. Then I must talk about the, I have already shown you this. This is actually Manfred Eigen, very old photographs. And this is two other persons. They actually discovered the flash photolysis. And again, you know that if you understand, it is very simple. Again, there is a reaction vessel. Everywhere there is a reaction vessels. So reactants are premixed and flow to the, and a very high pulse of light, very high energy. And then, of course, they produce some transient species, because most of the photochemical reactions are very fast, and their concentration is automatically monitored with the help of an instrument. So this is about this 1967. I am talking about the 1967. 1967, first Nobel Prize on the first reaction, water reactions. Formation of water. This is photochemical. Now, second Nobel Prize, 1999. A great and great scientist. Many times I always consider he is a great scientist in the case of the chemical kinetics, but it is in 1999. He is no more. He is an Egyptian scientist, but worked as a California Institute of Technology, and basically his reaction is the femtosecond spectroscopy. So therefore, sometimes it is called the transition state spectroscopy, but in this fast reaction technique, we will not study this femtosecond spectroscopy, but when we talk about the molecular reaction dynamics, then of course we will discuss about the contribution of the hammer as well. But basically it is also fast reaction technique, and this is actually called femtosecond spectroscopy, 10 to the power minus 15. 10 to the power minus 10 to the power minus 15. And I have taken this article from, as I told you many times, Journal of Chemical Education, one of the most important journal. You see that water reactions that is given in this Journal of Chemical Education. The whole issue is based on this formation of water. And then in our Indian journals also they have given this. Now the last example of fast reactions is a very recent article. It's October 2023, because you must know that while studying the fast reaction, femtosecond is now old, attosecond spectroscopy, attosecond, and it's great scientists, Krause, Willier and Agostini, left and right, they are all from Ohio State University, some are Lund University, Sweden. So they have been awarded this Nobel Prize, physics of course, in 2023 for this attosecond spectroscopy. So now you can understand their contribution, what is actually attosecond spectroscopy. Now 10 to the power 18 is not to the power minus 18, you see. We have started from 10 to the power minus 15, 1999, 2023, and 1967, starting from water. So this is the importance of that fast reactions. So just history, then I will start the stop flow method and the fast flow. Thank you very much. मैं उसको अलग